Let's talk about the central dogma and transcription. So the central dogma says that DNA can turn into RNA, which can then synthesize proteins. And the first step is called transcription. And then turning RNA to protein is translation. So transcription is the process by which we create RNA from DNA. And this is important because we must first convert DNA to RNA in order to then create proteins. So the first step to creating proteins is transcription. Transcription occurs in the nucleus of the cell. Let's now talk about the structure of DNA. So DNA holds genetic information. It's a double helix. And you should ask yourself, why is it double-stranded? Why not single-stranded? Well, because molecules go to their most stable form, and DNA is most stable as a double-stranded molecule. Let's look at this DNA structure. The DNA backbone is made up of sugar and phosphate, and they are bonded together by phosphodiester bonds. DNA strands are also anti-parallel. That means that one strand runs 3' to 5', while the other strand runs 5' to 3'. The 5' end has a phosphate, and the 3' end has the sugar. DNA also consists of different bases: adenine, thymine, cytosine, and guanine. Adenine and thymine base pair together, while guanine and cytosine base pair together. Adenine and guanine are purines. This means that they consist of 2 rings. On the other hand, cytosine and thymine are pyrimidines. They consist of 1 ring. You can remember this by thinking of the phrase pure as gold. One purine must pair with 1 pyrimidine in order to create the proper structure. If 2 purines pair together, or if 2 pyrimidines pair together, then the backbone will be bulky and misshaped. The bases are hydrogen bonded together. Adenine and thymine have 2 hydrogen bonds, while guanine and cytosine have 3 hydrogen bonds. The number of adenine equals the number of thymines, and the number of guanines equals the number of cytosines. A couple of more important vocabulary words are that a base plus a sugar bonded together is a nucleoside. Also, a base plus a sugar plus a phosphate group bonded together is a nucleotide. Let's now compare DNA and RNA. So DNA is a more stable molecule than RNA because it holds genetic information. Also, DNA is double-stranded, but RNA is single-stranded. This means that RNA does not have hydrogen bonding. DNA has the bases adenine, thymine, guanine, and cytosine, while RNA has adenine, uracil, cytosine, and guanine. DNA has the sugar called deoxyribose, while RNA consists of the sugar ribose. And finally, DNA tends to be longer than RNA because RNA does not have introns. Let's now look at major and minor grooves. So this is a major groove. while this would be a minor groove. A minor groove is the shorter gap on the outside of the DNA duplex. A major groove is the larger gap on the outside of the DNA duplex. These are important because transcription factors and proteins that we will talk about later use these grooves to recognize sequences on the DNA. Now, how would we write a complementary sequence of DNA? So the first step is to remember that DNA is antiparallel. That means that one strand goes 5' to 3' and the other strand runs 3' to 5'. Also, given a sequence of DNA, always pair adenine with thymine and cytosine with guanine. Let's now look at this example. If this is our DNA sequence, remember it's antiparallel, so our complementary strand will be 3' to 5', and then match adenine with thymine and guanine with cytosine. So our complementary strand will look like this, and this is our DNA duplex. Keep in mind that these strands are not identical, but are complementary. Let's now look at gel electrophoresis. This is our gel, and remember that gel electrophoresis separates DNA strands based on the size of the fragments. It also moves from negative to positive because phosphate groups on DNA are negatively charged, so they're attracted to positive. The gel machine is filled with agarose gel, which has little pores or holes in it. The shorter fragment will travel farther because it can move faster through the gel. We can stain our DNA fragments to see them on the gel like this. It's clear that the longer the DNA fragment, the less it will travel, so it will be closer to the negatively charged side, while the shorter the fragment, the farther it will travel. Separating molecules in a gel and staining them is called blotting, so one acronym that will be helpful is snowdrop. A gel electrophoresis done with DNA is called southern blotting. Northern blotting is for RNA, and western blotting is with protein. Let's now dive into transcription. This is our DNA strand with several important components. Before the promoter is upstream, and everything after the promoter is downstream. Our promoter consists of a Tata box, which has multiple thymines and adenines. This is where RNA polymerase will attach in order to begin transcription. The enhancer increases transcription by recruiting transcription factors. This is where an activator will bind. Transcription factors are various proteins that must bind to the DNA before RNA polymerase can bind. In order for transcription to happen, RNA polymerase and transcription factors Both must be present. Once DNA unwinds, RNA polymerase is the enzyme that will use the DNA as a template to create a complementary RNA strand. The RNA polymerase will add the bases adenine, uracil, cytosine, and guanine. Transcription start site is indicated as +1. Exons are sequences of the DNA that will remain in our mature mRNA. These code for important information, and we will discuss this more in a bit. Introns are non-coding regions that will be spliced out or removed later by a protein complex called the spliceosome. The poly-A signal sequence is a sequence on the DNA that signals termination. It also signals the addition of a poly-A tail. The terminator is where RNA polymerase stops transcribing. The template strand of the DNA is the strand that RNA polymerase uses to create mRNA. The non-template strand is not used for transcription, but it is complementary to the template strand. Once transcription happens, the primary RNA transcript will consist of exons and introns. It will undergo RNA processing. In RNA processing, we will remove or splice out the introns because they are non-coding, so we do not want to create proteins with the non-coding regions. We will also add a 5' cap to the 5' end of the mRNA, and then a poly-A tail to the 3' end. The 5' cap will help ribosomes find which mRNA transcripts need to be translated. The 3' poly-A tail will enable the mRNA to leave the nucleus and enter the cytoplasm for translation. We also have 5' UTR and 3' UTR, which are untranslated regions. They will not be translated by the ribosomes to produce proteins. Once the RNA has been processed, it is known as the mature RNA transcript, or mRNA, because it consists only of exons, not introns. And ultimately, it's important to remember that the mRNA is shorter than DNA and pre-mRNA because it does not contain introns. I hope this helped you better understand DNA, the central dogma, and transcription.